Here's another example of what can be done with microbiome research in the bowel. We've been looking at different types of cancer in the bowel. One that's very aggressive and one that's a little less aggressive. The one that's less aggressive is in this area here on the left side of the colon, coming down into the rectum. That's classical. And this form here, which is called SIMP, which is a special abnormality that's genetically based. And we find that there are different bacteria that are present in association with those two tumors. And it's important because the ones that are associated with a more aggressive tumor don't produce butyrate. And butyrate is important because it induces cell death, it prevents the proliferation of cells, it induces differentiation of cells. So perhaps in some forms of colon cancer, we will ultimately learn, as we're beginning to learn in a number of diseases, where modulation of fecal flora by using fecal transplants or probiotics will allow us to address these problems. Another example here, we're looking at HIV transmission. People don't talk very much about vaginal microflora. I think it's extremely important. We have a group of women who've been treated with antiretroviral gels. They nonetheless have high levels of inflammatory molecules in vaginal fluids. They're at increased risk for HIV. They too have different types of bacteria in their vaginal flora than women who are less susceptible and have less inflammation. And again, by targeting these bacteria, we can perhaps decrease the risk of infection with HIV. Now, there are a number of brain disorders that are associated with abnormality of the immune system. I showed you an animal model of one using that mouse that was exposed to group A beta-glutamate strep, a very common bacterium in the world, but in the inappropriate, unfortunate genetic context results in this autoimmune disorder. But we have narcolepsy and schizophrenia and all sorts of disorders, so this is not unprecedented. And there have been a wide range of viruses and bacteria and fungi and parasites implicated in inducing these immune responses. We can actually replicate some of these findings using not an infectious agent at all, but instead just a product of an infectious agent, like a genetic fragment that's double-stranded, which triggers innate immunity. The innate immune system goes all the way back to insects. Or lipopolysaccharide, which is the equivalent in bacteria. And we can then look at ME/CFS, and we find that there are a whole host of studies now that indicate autoimmunity, at least in some patients with CFS-ME, is quite prominent. So there's reason to think that there's going to be something there that we can do at the end by thinking in these terms. We also know that intestinal microbes modulate the immune system. This could be very, very important in finding ways in which we can not only understand the way disorders like MS, or schizophrenia, or autism, or CFS-ME may be triggered, but how that cycle of inflammation might be continued. So this is one of these. So I'm trying to give you the framework for why we think it's important to do that sort of work. We have been funded by a large number of people under the auspices of the Chronic Fatigue Initiative. It's really the major funders for that has been Graham Hutchins, who some of you may know. He had a very specific series of projects he wanted to pursue. We also received some support from the Evans Foundation. I'll be describing for you now some of that work, though some of you probably know us already. We have a study that comprises 200 cases and 200 controls. This has been added to the cases and controls that we already have from the CFS study that is focused on XMR, VP, and LV. We have also been able to look at spinal fluid, and we'll see why that's important in a moment, because I think that that's critical. The clinicians involved with that particular group asked us to evaluate a wide range of viruses and bacteria. There's a list here of those. And we have studied those in serum. And alas, looking at serum, we only found 2 cases out of 265 where HHV6 was present. And I know that Connie Knox and other people have been very invested in thinking about HHV6. And Jose Montoya at Stanford has done it as well. But my feeling is that some of those are selected populations where people have already been diagnosed with a herpes virus infection, and they wind up in that particular location. And as a result of a somewhat higher level. But if you look overall at all comers across these different programs, you generally find a lot of evidence for this, at least in plasma. And that's not to say it won't be present in another body site or in peripheral blood mononuclear cells, which are also circulating in blood, and that's something, in fact, that we need to do as part of microbiome study. With Jose Montoya, we then began to look at plasma again, and here's an example of his work. The other thing that we examined using high-throughput sequencing is whether or not there were other viruses that we would find that would not be present. Retroviruses proved to be fairly similar representation in the cases and in the controls, and we found a lot of what are known as anelloviruses. Now, typically, anelloviruses, and nobody really knows what they do. Anelloviruses are decreased or present in increased concentrations when people are immunosuppressed. What we found here, as you can see, in the cases, 22% had this alpha-protovirus versus 41% of controls. And similarly, as you go down through the beta and gamma protoviruses, people with CFSME have lower levels of these viruses, not higher levels. What the focus of our research at present now is to begin to try to characterize the gastrointestinal microbiome so that we can understand what bacteria, what fungi, what viruses are present in the key body compartments: the gastrointestinal tract, the vagina, peripheral blood mononuclear cells. We can do metabolomics, identify products of bacteria that might be important in causing dysfunction. We can study the proteins that are present in the blood. We can study the RNAs that are present in tissues and in blood, so we can get insights into the genetic predisposition to an abnormal response. And we can then begin to link this with functional immunology. Now, the last thing I simply want to say is that, we have a very large group, it's some 60 individuals in the U.S. and many, many more around the world. We focus, and you will chiefly see my name in association with things like Ebola and MERS and SARS and things like this, right? And I am very committed to working with our Centers for Disease Control and the World Health Organization in addressing those challenges. But I think that the future of microbiology and immunology really rests with understanding the role of microbes and immune responses to microbes and toxicology in chronic diseases. And I've devoted my career to thinking about things like depression, schizophrenia, autism, and more recently, a large focus of our research has been on CFSME, because I think that this is, as I've shown you, since 1997, this was something that we wanted to do. We think the tide is finally changing, and I think it's people like you who are in a position to influence your policymakers and say, we want you to address this problem, we're hurting, and so forth. As you all know, within the UK, data are fairly similar in the United States. 1 in 250 to 1 in 500 people has CFSME. The cost estimates in the United States, and we have to use financial estimates in these days when it's very, very difficult, is that the direct and indirect expenses associated with CFSME in our country, roughly 320 million people, is in excess of $20 billion per year. So it should be possible to make, I think yours is something like 6 or 7 billion US dollars, which I guess is what, 4 or 5 million pounds. So I think you can make this argument both on a humanitarian and compassionate basis and on an economic basis. And I would encourage you to do that, because that is what succeeded for us with many of these other disorders, whether it's the March of Dimes, polio, or multiple sclerosis society, or Alzheimer's, what have you. You have to be as vocal, you have to be as organized, and you have to be as focused on your mission. Thank you very much.